area about 2000 when I started reading a lot of information on what was then a new medium, the internet. And what really bothered me was at least 95% of the information I read on the internet about GM crops was inaccurate, misleading, or blatantly false. And so I started to research the subject in great detail. Today it's a little different. Today there's far more accurate information out there on the internet. I would say probably about 60-40 for the false information, and they're still winning the web pages numbers. But a person who wants to find out good, accurate information on this particular subject can use the internet if you're careful about sourcing. So we're going to talk about genetically modified foods, and hopefully I can give you some context about where it fits in the role of global agriculture production. The first thing you have to know is there's nothing natural about agriculture. Agriculture is the manipulation of DNA of plants and animals to suit our needs. It has nothing to do with the benefits of our food. We have been changing the DNA of our food for 10,000 years. The only thing that is different with GM crops are the methods we use to change the DNA. In 1987, the National Academy of Science in the US stated very clearly that there was no evidence of unique risks from recombinant DNA and the movement of genes between species or genera. If you jump ahead 25 years of research looking into those very questions, it's the exact same opinion. So if we look at how we have produced food for the millennia, we start with some weeds here. Anybody want to guess what those weeds have become by changing the DNA? Any guesses?
Carrots.
Good. The left became carrots. By changing the DNA of that wild plant, we got carrots. The middle one, by changing the DNA, we got lettuce. And the right one is teosinte, as you mentioned. It grows in the Mexico region of Central America. And as you can see, there's very large differences in the phenotypes of these plants. The reason they're different is because we've changed the DNA. Here's a modern day tomato. Nice, big, red, juicy, tastes like cardboard. Sorry about that. But if you look at the ancient varieties of tomatoes, they're small and they're toxic. They produce large amounts of glycoalkaloids that make them inedible. It's by changing the DNA of these wild plants that we got to today's modern day tomato. Potatoes are also a member of the same nightshade family, also produce a variety of poisonous glycoalkaloids. Here's many different varieties of wild potatoes. And by changing the DNA of these wild potatoes, downregulating or removing the glycoalkaloid proteins, we get the modern day russet potato. What's interesting is right now in the UK, there are field trials going on looking at blight-resistant potatoes. So most of you have heard of the Irish potato blight. A particular pathogen, fungal pathogen, that wiped out the potatoes back then is still present today. It does $2 billion of damage to potatoes around the world. Researchers found some genes in those wild potatoes that conferred resistance to the fungal pathogen. They transferred them into the commercial varieties of potatoes and were showing tremendous success. The critics didn't like that. They tore up the fields because we put potato genes in potatoes. It's a little difficult to understand that logic. This is a wild brassica, probably the most manipulated plant in food production. By changing the DNA of the brassica at 500 BC, we got kale. By changing the DNA of kale, we got cabbage and kohlrabi. By changing the DNA of cabbage, we got cauliflower. By changing the DNA of cauliflower, we got broccoli. Now, all of this was fine and good. Then science went off the rail. They produced Brussels sprouts. Sorry about that. But as you can see, from here all the way down to here, very large differences in what the vegetable is. It didn't happen by chance. It happened by man or humankind manipulating the DNA over the centuries. That's what agriculture is. In the last 80 or 100 years, we've used a variety of artificial methods to also randomly change the DNA. This is a perfect example. This is a radiation breeding facility. This particular one is in Japan, but they exist around the world. It's about 600 feet across, and it's surrounded by a 25-foot-high cement wall. And as you can see, all the different rows of crops being grown here. Once they're near ready to be harvested, right in the middle there is a tower with a cobalt-60 radiation source. So everybody gets out, they drop the shielding, and everything in the 600 diameter is irradiated. They then put the shielding back up. Massive changes happen to the DNA. Huge changes. Most of the time, the seeds that are harvested die. They do not replicate. But every once in a while, a seed will replicate. It will grow. They'll then take those surviving seeds and grow them up to see if we got any trait we like. Does it give us a redder tomato? Does it give us a disease-resistant variety of wheat? If it does, it gets shuffled into the agronomic system to determine whether it will become the next cultivar in agriculture. Nobody has any idea what we've done to the DNA, how many changes, where the changes are done. None of that is ever recognized. We've been using this for about 80 years. There are 3,000 different varieties of crops that have been commercialized using this technique. The only reason we know that is because the IAEA monitors all atomic energy usage around the world. Thousands and thousands more crops have been produced using chemical mutagenesis. We don't track those. Same idea. Random mutations of the DNA, absolutely no idea what we've done. We've just got a product we like. That's how we've been breeding crops for the last half-century and more. If you compare that to genetically engineered crops, where we take the exact gene for the exact trait we want, we place it into the crop, we know exactly where it's placed, we know exactly how it's expressed, we know exactly how it affects the genes around it, if at all, we know exactly how it affects the plant as a whole, we know how it's expressed in every single tissue, we know how it affects the plant's interactions with the environment around it. All of that must be known for every GM crop before it's allowed to be commercialized. That's part of the testing process. So if you look at conventional or traditional breeding, we mix thousands of genes, we mutate unknown numbers of genes, and we have cases of extreme inbreeding, or where we force two entirely different species together to make new crops. That's all fine. Nobody worries about that. Add one or two genes, knowing exactly where they're from, what they do, where they are, and we have decades of testing that has to be done. No GM crop is ever brought to market without very extensive testing, and it costs a great deal of money. Now I'd like to talk about the power of one gene. On the right is a variety of papaya, rainbow papaya, and on the left is a transgenic variety of that papaya. One gene was added. The gene was from the coat protein of a viral pathogen that is devastating to papaya plants. It's called the papaya ring spot virus. By taking just the one gene for the coprotein and putting it into this plant, you get this plant. These are both grown in the presence of the virus. The transgenic is immune to the virus. This work was done at Cornell University in the late 80s, and by 1991 they had a very good collection of transgenic crops ready to go. They knew this was going to be important because in 1994 the papaya ring spot virus showed up in the Puna region of Hawaii. It decimated the papaya plants. But fortunately this work had already been done. There were a great many of the transgenic plants ready to be seeded. So the work began quickly. Here you see 13 months after trials, transgenic, non-transgenic, planted at the same time. They're planted in the environment where the virus is present. We had 17 months, transgenic, non-transgenic. You can see it's really starting to show the difference. And if you look from above, it's very clear. The only difference between these on the outside and these on the inside is one gene. The gene is present because the virus that's doing all this damage out here has a gene as well. There you see 1999, nice, very good yields. Plantations are quite productive in the presence of the virus. And if you think it changed, here's 2011. In the front, non-transgenic, and behind it, the transgenic. The virus is still present in the Puna region of Hawaii, still decimating non-transgenic papaya plants. And in fact, 85% of all papaya grown in Hawaii is now transgenic. And some of the people who want to grow non-transgenic surround their fields of non-transgenic with transgenic to protect them. Interesting. This represents a cartoon of the genetic structure of the most popular rice grown on the planet, IR64. So starting back here, this blue represents the ancestral genomic structure of the predecessor to IR64. Everywhere you see yellow is a mutation event, so that could be chemical or radiation induced. Everywhere you see a pink is a recombination event where other genome material is brought in from other species. Everywhere you see a dark blue is a translocation event where DNA is moved from one chromosome to another. And everywhere you see a gray, or part of the DNA, has been removed. This is where we started. This is IR64. Very little of the DNA of the ancestral genome has not been manipulated on a very large scale. Nobody batted an eye. Now they added 3 genes. They added 3 genes that allow this rice to produce beta-carotene. Anybody see where the 3 genes are? Anybody see that? It's actually right there. That little pink in the middle of that blue streak. 3 genes were added. 15 years later, it still has not been commercialized. They are still testing it. From here to here, no problem. Add 3 genes. Oh no. There's golden rice. 1 billion people on the planet suffer from vitamin A deficiency. Golden rice will help those people. A 100-gram serving gives you 50% of the daily RDA of beta-carotene, which our bodies then convert into vitamin A. Hopefully next year, it will be finally given away to subsistence farmers in the Far East, where the greatest need occurs. So, one of the questions you often, or one of the concerns you often hear, is GM food safe? Let's look at that for a minute. If you look at the way it's tested for food safety, these are the European Food Safety Guidelines for risk assessment for GM crops as of a couple of years ago. Principles of risk, there's 4 subcategories. Molecular characterization, so that's where the DNA is, how it's expressed in each tissue, how it's expressed, how does it affect the genes around it, all of that. Comparative analysis, how does it compare against the parent crop, or what's known as the isoline. the genomics before the transgenes were inserted. Toxicology, 5 categories. Allergenicity, 3 subcategories. GM crops are the only crops on the planet that are tested for potential allergens before they're commercialized. No other crop on the planet is tested at all for this issue. Nutritional assessments. All of these categories and subcategories have multiple different tests associated with them. So as you can see, the extensive testing program is the reality. People will often say GM crops are not tested. On the right is a folder of a variety of flax. It's allowed to be grown, all the tests that had to be done to grow across North America, Canada and the US. They added 1 herbicide tolerance gene. On the left are all the tests that had to have been done to allow that now transgenic variety of flax to be sold in Canada. Didn't even allow it to move across the border into the US. You can see that testing is very, very extensive. The next time somebody tells you it's not tested, ask them, what test, not already done, would you like to see done? I've never got a good response. People often think that Europe science has a different opinion of GM crops from North America. That's not true. European politics, absolutely. This document, 2011, A Decade of EU-Funded GMO Research, makes it very clear, after 130 research projects, 25 years, and 500 independent researchers, that GM crops are not per se more risky than crops from conventional breeding.
Sorry, could I just ask you where this came from?
A Decade of EU-Funded GMO Research.
Yeah, but I mean, what organization?
European Commission. The link is on my website. The European Academy's Science Advisory Council. These are members of every European's National Academy of Science. This document came out in 2013, the future, or Planting for the Future. There is no valid evidence that GM crops have greater adverse effects on the health and environment than any other technology used in plant breeding. They're pretty strong words, and that's from European science. The World Health Organization, and I recommend everybody Google 20 questions on GMOs. You'll get the World Health Organization document. This is one of the answers. I think this is question 12. GMO products that are currently on the international market have passed all risk assessments conducted by national authorities. Different assessments follow general basic principles, including environmental and human health risks. These assessments are thorough, and they have not indicated any risks to human health. That's on the 2013 WHO website. Last year, I spent a great deal of time over in Richmond talking to city council who were thinking about banning GM crops. They asked me to present the science to them. They also contacted the Vancouver Coastal Health Authority and asked their opinion. They didn't expect this response. There is no evidence that Health Canada approved GM crops in food are any less safe for human health than non-GE varieties, and there is no public health reason to ban GM crops. Richmond decided to disregard all the science that I presented to them. You've seen some of it, I'll show you some more. They chose to ban GM crops anyways. Symbolically, because it's not in their jurisdiction, it's actually regulated at a federal level. The American Association for the Advancement of Science in 2012 said that the World Health Organization, the American Medical Association, the US National Academy of Science, the British Royal Society, and every other respected organization that has examined the evidence has come to the same safe conclusion. My favorite statement from a large body is actually from Health Canada. The overwhelming body of scientific evidence continues to support the safe use of genetically modified foods and feed products in general. However, whenever new information comes up that may question that, it should be analyzed. I think that's a pretty sound policy. So we can see, if you look at global food safety experts, they're all in agreement. GM crops do not represent any unique risk. We've got 25 years of research, and we've got 3 trillion meals containing ingredients from GM crops. Not a single documented case of harm has ever been brought forward. That's a pretty good track record. Nothing is absolutely safe, but the track record of GM crops is impeccable. Let's look at the next issue. People often tell you that GM crops threaten organic agriculture. But organic agriculture is not certified based on the end product. Organic certification is based on the process used to grow the crops. The end product purity does not come into play for the certification of organic food. That's the Canadian, the US National Academy Program. Unintended presence of products of excluded methods should not affect the status of organic production. And if you want to go globally, the International Federation of Organic Agriculture movement says that the potential of GM contamination does not alter the traditional approach of certifying organic agriculture. It is a production method. The IFO does not support any threshold to be introduced for GM content, and they do not even support testing for GM content, trace amounts of GM content in organic food. You still hear many, many people tell you that organic food is threatened and has been damaged by GM crops. Last year, the Organic Seed Growers and Trade Association sued Monsanto, probably the largest corporation producing GM crops today, over exactly that. 30,000 plaintiffs. The judge said, please bring forward your evidence of people losing organic certification for trace amounts of GM crops showing up in the field. None showed up. Not one. Not one person had any evidence of that. The other thing they sued Monsanto for was that apparently, they claimed, Monsanto was suing farmers whose GM crops showed up in their non-GM fields. The judge asked, fine, bring forward that evidence. Not one person stepped forward again. The judge threw out the case saying it was a clear attempt to create a controversy where none exists. The appeal was denied, and just recently, last week, the Supreme Court of the United States turned down any further appeals. GM crops and organic agriculture have both coexisted very well in the last 20 years. Both have grown up to be very successful types of agriculture. GM crops do not threaten organic agriculture. Organic agriculture would be an ideal way of doing certain types of agriculture, except for the big problem. We've got 9 billion people coming. We need to produce 70% more food on the same land. Organic agriculture has a 30% yield drag on average to conventional agriculture. How could we possibly feed more people on the same land using less efficient methods? Another thing you often hear is that organic agriculture or GE agriculture is not sustainable. What does the science say about that? The National Academy of Sciences 2010 report, The Impact of Genetically Engineered Crops on Farm Sustainability in the United States. Their key finding, in general, the committee finds that genetically engineered technology has produced substantial net environmental and economic benefits for the farmers who grow those. What does the European science say? In that same document, a decade of EU-funded GMO research. Now, after 25 years of field trials without evidence of harm, fear continues to trigger the precautionary principle. Europe needs to abandon this knowingly one-sided stance and strike a balance between the advantages and disadvantages of technology based on the scientifically sound risk assessment process. They're still fighting it. Politically, scientifically, they're not together. So what are we talking about? The first generation of GM crops have been around for almost 20 years now. We're talking about three main characteristics. We've got herbicide tolerance, insect resistance, and viral resistance. I showed you one example of viral resistance with golden rice. Let's look at herbicide tolerance. By far, the most acreage grown on the world for a GM crop is herbicide tolerance, where the crop is engineered to be resistant to glyphosate, or what you commonly know as Roundup. Glyphosate was patented in 1973 by Monsanto. Patent has since expired. It's made by hundreds of companies around the world. It's a very simple molecule. You've got a glycine linked through a methyl group to a phosphate group. All three of these components are found in every living cell on the planet. They're just put together in a unique way. Glyphosate breaks down completely into non-toxic compounds in a relatively short period of time. Temperature, humidity varies that time. The EPA considers Roundup or glyphosate a level 3 or 4 toxicity rating. That's its lowest rating. The level 3 comes from one formulation of glyphosate or Roundup that has a surfactant in it that is harmful to fish. And there are rules. If you're spraying Roundup near bodies of water, you have to use a special formulation that doesn't have that problem. The EPA considers that's a national toxicology program. There is no evidence of any genetic or reproductive toxicity with glyphosate. Its skin irritation is equal to baby shampoo and better than Pine Sol or dish soap. And as for carcinogenicity, it has none. It is not a carcinogen. It has no effects on amphibian development. When used properly, it has no effects on soil, bacteria, and fungus. It has no effects on earthworms. It has no effects, again, used properly, with aquatic invertebrates. And we hear a lot lately about bees. It's non-toxic to bees. But what does it mean to us? We're the end users of GM crops and derived food. The National Institute of Health in 2005 did a very large study looking at 57,000 licensed pesticide applicators, their families, and they concluded that glyphosate exposure was not associated with cancer incidence overall or any most subtypes. There was one subtype that showed some kind of a linkage, so they investigated that some more. In 2011, the epidemiological study looked at non-cancer health outcomes. And our review found no evidence of consistent patterns of positive association indicating a causal relationship between any disease and exposure to glyphosate. In 2012, the same epidemiological study, this time about cancer specifically, and no consistent pattern of positive association. This is what the science says. Glyphosate has been studied for decades. It's no longer controlled by one company. It's made by companies around the world. And if you compare glyphosate against the compounds it replaced, the environmental impact quotient for glyphosate is far lower than the compounds that it replaced. Now switch gears to the insect resistance. Most of those traits rely on this particular bacterium, a soil bacterium found around the world, Bacillus thuringiensis. It produces a variety of proteins. I think the number is well over 40 now, different proteins, and each one of them targets a specific insect group or pest. The protein, once consumed by the pest, must find a receptor lining the gut of the pest insect. If it doesn't find that receptor, then the Bt protein is digested like every other protein, and that's us as well. If it does find the receptor specific for that particular protein, it then binds to it and produces a hole in the cell membrane. That causes the cell to die. Here's a few details about a few different Bt proteins. Bt1A has a no observable level of mortality. That means it's safe. You can't demonstrate any effect. 4,000 milligrams per kilogram. That's a sugar cube size piece per every kilogram, and it has no effect. Same rough areas for several other BTs. Notice here, table salt. It's LD50. That means 50% of the test population that are fed this level die. That number looks suspiciously like that number. Nobody's calling for the banning of table salt. In the US and in other parts of the world, 2/3 of the insecticide spray is on two crops, corn and cotton. Both of those are very much insect-resistant Bt crops today, and that has allowed hundreds of millions of pounds of broad-spectrum insecticides to not be used while the farmers maintain their yields. This is a UN OECD document, consensus document on BTs, and basically it says that there's negligible toxicity to humans, and when it comes to the environment, the overall safety record of BT has been established in laboratory and field studies when they looked at formulated BT sprays. That's what the organic agriculture people use. They use Bt as an insecticide. They use the whole live bacteria. Or in GM crops, where we take just the one active protein that actually does the killing and put that into the food crops. Safe. In North America, we've been using Bt for over 40 years. Before GM crops, it was in Bt sprays. Bt-type insecticides have been used in the Orient for over 100 years. Very long safety record for this particular type of natural insecticides. You see a lot today that Bt crops are responsible, or GM crops are responsible for the die-off of bees. We definitely have a problem with bees, but it's not anything to do with Bt. Scientific beekeeping says the verdict of Bt crops, the specific cry proteins used in GM crops, were intentionally chosen not to cause harm to bees. And there is no evidence to date that they do. On the other hand, Bt crops require far less insecticides, and we know insecticides have a nasty effect on bees, including organic pesticides, which have a very nasty effect on bees. And the USDA says that there is no correlation between GM crops in general and colony collapse disorder, citing areas where there is colony collapse disorder and no GM crops, or other areas where there's GM crops and no colony collapse disorder. This is what we've seen from the first 20 years of GM crops. We've had about an 8%, 9% drop in insecticide use. But more important is the environmental impact, what's known as the EIQ, the environmental impact quotient. We've had a 17% drop in the environmental impact at the same time as we've increased yields. In Australia, 95% of the cotton grown is Bt, and that allowed for an 85% reduction in insecticide use. In India, they first commercialized Bt cotton in 2002. Since then, we've got a 24% increase in cotton yields per acre, a 50% increase in profits to the small stakeholders because they don't have to buy insecticides. And India as a country has more than doubled its yield of cotton since 2002. It's no wonder at all why over 90% of the cotton grown in India today is BT. People will tell you GM crops do not yield well. They haven't looked at the data. This paper was